The dumbest part about this conspiracy theory is that if you want to kill off U.S. science, just elect
Exactly.
Donald Trump.
Exactly.
Like, and get Russell vote into. Like,
You've done it. You've already did it. You don't have to worry about our well-being too.
This is Audio Immunity, a podcast about our body's never-ending fight with the outside world.
hello, everyone. This is Audio Immunity, episode 55, recorded on April 22nd, 2026. I'm Kevin Bonham, joining you today from my home because it's April vacation week.
Woo!
For those of you on video, forgive the pink light in the background, trying to bring my seeds up. And I'm joined, as ever, by Matt Woodruff. Hello, Matt. Had to do 3 takes on that.
That was
but
the best.
it's
That
fine.
was the best of the 3 takes. Yeah, I
Thank
think you
you.
should leave all of them in. Yeah,
Yeah, I'm going to do some judicious cutting.
fair enough.
I was just
Hi,
saying,
everyone.
I've got a pink light behind me. I've got seedlings. It is still too cold in Massachusetts to put them out because who knows when we're going to have another frost. But things are, last time we talked, I was saying how gorgeous the weather was. And then for the last 2 weeks, it's been in the 40s and 50s
Yeah.
and sometimes frost overnight. So
Yeah, it actually got
New
chilly
England,
here
man.
too. Chilly means a different thing, I understand, in Atlanta versus Boston.
Yeah.
My son goes out at 6. What do we do? We do 6:50 is his bus pickup time, and so we have to, leave the house around 6:40 or so and walk over there. And it has definitely been jacket weather for the last couple weeks. So actually, I think starting to turn today, I think it's supposed to get up to 75, 80.
Okay. Well,
I
that'll be
once
nice.
again plug for Atlanta is hotter than Boston. Now that's going to
Always
turn,
true. Almost
as
always
you
true.
know. Yeah, yes, exactly. Highly reproducible, especially when Atlanta's 108 with 115 in humidity, and we'll get there in, I imagine, 6 days. So
Yeah. Good luck to you. Today, we are discussing a very cool paper that we'll get into in just a sec. But I think, let me just introduce the paper. So the title of the paper is Neutrophils Self-Limit Swarming to Contain Bacterial Growth in Vivo. And the last author is Tim Lamerman. First author, Corbinin Kienel, maybe? Kienle? I have no idea. They're from Freiburg in Germany. And the main thing I wanted to say as just an intro to this paper is that this is one of the most beautiful papers visually that I have seen in a very long time. It's just lovely. There's great diagrams.
Mhm.
There are embedded into the PDF are links. I mean, they're links because you can't do videos in movies, but there's not only figure 1, figure 2, there's also video 1, video 2. And I'm used to those being thrown in the supplement, but here they're taking up giant chunks of real estate on the page itself. It'd be interesting to see what it looks like in the print version of this article. It is published in Science.
Yeah.
So what, we're going to get into neutrophils, which we've talked about before. We're going to get into swarming. Lots of very cool stuff about this paper. But do you agree with me? I feel like if I have one paper that is this visually beautiful in my career, I will be happy. It's just very lovely.
See, this felt like a trip home to me. Yeah, it is. It's very pretty. And we talked, and part of the reason that I selected this paper actually was because last time, and this is a plug to go back one episode, last time we talked a little bit about microscopy and the spaces where microscopy has really developed. And specifically, we talked a lot about spatial transcriptomics, which is the assessment of the full transcriptome or partial transcriptome of individual cells on a slide using a microscope, right? So you're basically investigating gene expression in situ, right, in space. But we talked about kind of the progression of microscopy that got us to that place where we were able to to actually visualize some of those things. And one of the directions we kind of wandered down was different iterations of fluorescence microscopy. And so, we talked a little bit about the kinds of technology that spatial transcriptomics relies on, which is the use of lasers, where you essentially take a slide, lasers, and you
Was that inflection really necessary?
yes, it is.
Lasers.
Lasers. And so we put tissue on our slide and we stain that tissue with protein-specific antibodies, and then those antibodies carry fluorophores, right? And then you take a laser and you go pixel by pixel and you shoot a laser at that space, and if there is a fluorophore there that gets excited by the laser, the fluorophore releases a photon in whatever wavelength it likes to do based on who it is. And then you capture that. And so that is confocal microscopy. It's been a really standard way of visualizing for a long time now. And there are ways of doing confocal microscopy that allow you to actually track live cell fluorescence. And so we're going to see a couple examples in this paper where we're actually tracking individual fluorescently labeled cells as they move around in a dish. And I think it's a spinning disk. I would assume that it's a spinning disk. I didn't actually look in the methods. But also what we're going to look at here today is an application of that in vivo. And this is, what I spent a lot of my time doing in graduate school, which is essentially instead of shooting one laser at full strength, right, in one single plane, what you do is you fire multiple photons at partial strength from different angles. And so what you're doing is, and if you're watching on YouTube, you can sort of see my hands. What you're doing is you're firing those photons from different angles so that they hit each other at a specific depth within the tissue, right? So what you're doing essentially is, instead of shooting a laser that fires straight through the tissue and excites all Z planes within that XY coordinate, you are also hitting specificity on the Z plane as well. So you're essentially exciting only one point in space, right, at full strength. So, this was appealing to me just because, we had kind of introduced some of those concepts. Go back again for that discussion. I think it was pretty good, actually. And then here we're using that advance in technology to really understand something about how the biology of these cells changes their migration patterns, mostly in vitro, but also we have some in vivo examples as well. And I'll point those out when we get there. But as I also mentioned in the previous episode, this is what I spent a lot of my time doing in graduate school, and so these tracer plots and the quantitations and all of that actually felt very, very familiar. And yeah. Anyway, there was a reason that my brain, I think, stuck to this. It's like,
Yeah,
oh,
well, and this is
look,
a fairly old paper now.
it is.
I mean, old as these things go. It's from
Yeah.
2021.
Yeah.
Which I actually didn't realize until quite late in the game. But yeah, I think so. The basic concept here is around these cells called neutrophils, which we've definitely talked about before. We've talked about their crazy death strategy, neutrophil extracellular traps, where they'll like eject their DNA into the into the extracellular space to trap bacteria. I don't think we've talked about this swarming behavior, except maybe indirectly before.
Yeah.
So I do remember from graduate school seeing videos of neutrophil swarming, I think in the context of intravital imaging. But basically, you have these lovely videos where you just see like a point, imagine a point in the middle. And I think for those of you watching on video, I'm going to do a little bit of extra editing and I think splice in the actual videos as we're talking about them here. But
Fancy.
we're just going to have like imagine a point in space and a bunch of green little dots that all just like swarm and aggregate right at the same spot, or come from one direction and just come en masse to another direction. It's like a bunch of bugs like crawling towards something juicy. It's very, very cool to see. And this paper does a bunch of quantification, but specifically they're looking at a the way that they are communicating with each other rather than sort of focusing on individual cells and how they are swarming towards something like a microbe. They discuss this sort of communication between neutrophils that controls their swarming behavior, which I hadn't known about. I mean, like I said, this is a 5-year-old paper, but it had not come into my consciousness before that this was a thing. So I wonder if we want to talk a little bit about GPCRs at all, or if because the actual receptor that is guiding, one sec, delivery. So the actual receptor that is guiding both the sort of movement of the neutrophils as well as this swarming behavior is a G protein coupled receptor, which is one of these things that you learn in cell biology. I feel like, at least I learned it in cell biology in undergrad, and then mostly haven't really thought about. It's one of the sort of classes of receptor activation sort of paradigms that is quite cool. It involves the receptor binding and then enzymatically modifying another component of the membrane, which then sort of transduces the signal. I don't know if any of that detail is actually important. You can go open up your Albert's cell biology textbook if you want to learn about this in detail.
Or ask Google or ChatGPT.
Or ask Google, ChatGPT.
Yeah.
That's true. That's true. They will describe it. Although
They
I think
will.
the pictures will not be quite as nice.
That is true. 7
but-
transmembranes. That is the
That's
only
true.
thing that I recall. Just like if I think about it, I could probably pull more, but 7 transmembranes is the thing that immediately pops to my head.
Yep, absolutely. That is a key component. Which incidentally always confused me because like there doesn't seem to be anything intrinsic about the 7 transmembranes. Maybe there is, but I don't know. Anyway, we're going to talk in this paper mostly about this GPCR kinase. So an enzyme that phosphorylates a GPCR called GRK2. And I think maybe we should just get into the first figure, right? They're mostly, this entire paper is basically, they wanted to look at these GRKs and their role in neutrophils. And so they do a bunch of knockouts and they find out that this GRK2 is cool. And that's basically the whole paper, is digging into the different ways that GRK2 is important.
Yeah, that is functionally the case. Yeah. I would say, one thing that we want to distinguish between is the signals that are being used here in this paper to assess the swarming behavior, which is this communication behavior between neutrophils, and also the initial recruitment signals, right? So just some, getting back to some really, really basic neutrophil biology. So neutrophils are the most abundant kind of white blood cell in your blood. So they are constantly being called into local environments to, most of the time, just clear minor incursions, right? So these are sort of your troops on the ground, earliest responders, except for obviously the tissue resident populations that are already there. And so these guys essentially start in the blood. They wait to see some sort of activating inflammatory signal out in the periphery. Alongside, and we've talked about this before, alongside many things that accompany inflammation at the local tissue sites, a couple things happen. One is that you activate the local vasculature. And when I say activate, what I mean is the local vasculature. So blood endothelial cells, for example, will start to express different kinds of adhesion molecules that they weren't expressing before. And also the really tight junctions that blood vessels need to continuously maintain with each other, these endothelial barriers, tight junctions, they need to relax a little bit, right? Because if you're going to use the blood in order to recruit cells out into the periphery, you need you need a way for them to get in. So essentially what's happening is these neutrophils are flying along in blood vessels, just kind of hanging out, not really doing all that much. They become exposed to chemokines or chemotactic agents in specific areas. So imagine you're flying along in a capillary, and all of a sudden you start to sense some chemokine in the local environment. At the same time, you stick to the vessel wall because there are some new adhesion molecules that are allowing you to roll along that vessel, so that when you encounter that chemokine that you notice, you activate your "and I'm here" signal. And those are integrin activation events usually. And that allows the neutrophil to essentially migrate through the junctions between endothelial cells in the blood vessel and find its way into the inflammatory tissue, right? So a
I have a
neutrophil
question for
doesn't.
you.
Yes.
I have a question for you about this. So we spent a lot of time in our immunology class in graduate school learning about this process because
Rolling adhesion.
Uli and his descendants, a
Yes.
lot of them were where we were at. So this is Uli von Andrian. Do most people, like, does your immunology graduate program talk a lot about this stuff?
Not as much as I remember us talking about it, and
Okay.
at some level, to their detriment. I spend a lot of time talking about this stuff, about spatial context of immune responses and like how we organize things. Uh, yeah, I think without there is a lack of understanding as to how spatial regulation happens. And, to be fair, I'm that faculty member. So, there really isn't a huge gap in the program because I'm there talking to them about it. But I would say that there isn't a ton of like inside out signaling. I don't know if
Yeah.
you remember, but like Tim Springer
Yeah.
gave this lecture over and over again where he did this and then he
Yeah.
did this.
Yeah, no, I
and
remember it very clearly. There's like this 3-component address system. I think about phone area codes and all of this stuff.
Exactly. And
And
I do think
it feels like it looms very large. I'm not involved enough in the Tufts immunology graduate program yet, so I don't really know what the scope of the training is here, but I feel like it may be the case that we got a much more in-depth uh, education on this aspect of immunology than most people tend to get,
that
just
it
because of
could
who
well
was
be.
there
Yeah,
when
it,
we
it
were in
could well
graduate
be. And I
school.
think that honestly, that is a feature. And it took me a long time to recognize this. And maybe it took me until I was a faculty member at Emory to recognize this. But your program really is just made up of the faculty that occupy the research space, right? And so everybody can go through Janeway, and people do at some level. Um, but you're highlighting a lot of like classic immunology there, right? You're going to get a lot of
Hmm.
VDJ recombination. You're going to get a lot of, ontogeny and, innate versus adaptive and like all of that stuff. But the sort of specialized flavor of these programs really is made up just of the faculty
Yeah.
members and what their interests are and what they happen to show up that day. Because that's the other thing that happens that scares me a little bit is you'll show up to a lecture and you'll be like, I forgot to talk about. And then
Yeah,
you
right.
realize that thing you forgot to talk about might never be talked about in the entire career of that graduate student. So it's, yeah, it's an interesting thing being on the other side. But I say all of that to say that all of those signals, everything that we just talked about to get a neutrophil, which is flying down a vessel at hundreds of body lengths per second, um, none of that is related to what we're talking about today. So, so
Right.
all of those signals are very important, and all of those chemotactic signals are coming from cells that are already in the tissue, right? And so that's a really important distinction. So those cells will eventually find their way into the tissue. They will find their way to the source. And what we are talking about here are signals that initiate from the neutrophils themselves and
Well,
signal
yeah,
to other neutrophils, right?
there's a combination, right? So what Matt was just describing is the inflammatory environment that gets neutrophils from the blood into the tissue. And they're going to recruit, like Matt said, neutrophils, major component of blood vessels. They're coming in like shock troops. As soon as there's inflammation, they're coming into the tissue. Once they're in the tissue, they have to find their way to the actual thing that's a problem. Sometimes that's going to be coming from cells, but most of the time that's going to be neutrophils with their own pattern recognition receptors and their own sort of chemokine sensors finding their way to the microbial products. But then there's this additional piece now that is coordinating. You have a flood of neutrophils get into the tissue. All of these neutrophils then need to go within the tissue to find the source of infection or damage. So sometimes it's going to be cell-produced molecules in the case of injury, which we'll see later. But then, so there's the getting into the tissue, there's the locating the source of the problem within the tissue, and then there's this third thing, which is what this paper is about. That is
and
how the neutrophils are sort of coordinating with one another.
Yeah, and I would say too, in addition to that, and I think it's sort of a midway layer there, that it's been known, and they talk a little bit about it in the discussion, that there is this sort of layered approach that neutrophils rely on. So it's known that autocrine signaling, right, this sort of self-produced CXCL2, LTB4, that sort of signaling process is obviously necessary. But if you get sort of one of these outside signals, it trumps the autocrine signaling, right? So if you have like C5a, right, which is a highly chemotactically active complement derivative, right? So these are the proteins that just sort of hang out in your blood and they randomly attack stuff. And unless you have the machinery to fend off the attack, essentially, you are now like stuck to these proteins that start releasing chemotactic factors, right? So if a neutrophil comes into a tissue and it's seeing the autocrine signals, it's seeing CXCL2 and LTB4 produced by another neutrophil, and at the same time it sees C5a, it goes and finds C5a, right? Because that is sort of the initiating inflammatory system. But yeah, and you're absolutely right. It's like there's this push-pull of neutrophils needing to find each other, but also needing to be able to continuously respond to other cell types and other locations that might be able to direct them to wherever the next site of infection is, right? And I think that is, at the end of the day, the tension that this paper is really trying to resolve. Like, how does all of that complexity actually play out?
Yeah,
And
an analogy just occurred to me, which is like neutrophils are the mob, right? I mean like a mob of not the mob like the family,
mob.
like a mob, mob dynamics, right?
You
So
come to me in the day of your infection.
if you are a specialized like, let's say, like, thinking about crowd dynamics, if you're like a police officer or something, you might have like a specific place you're going to go, you're going to look for specific signs. But if you're just following the mob, you're going to just like, hey, there's a crowd going this way, I'm going to go that way, because that seems like where the action is. If you then see something specific, you might then sort of turn from the mob behavior. But yeah, neutrophils are, like, I think of them like the berserkers of the immune system. I don't know if you ever did like role-playing games or whatever, but they're basically just like, they go in and they just start smashing shit. That's their role.
Yeah, although I will say, and this is, again, one of those features, because this is an area that I think, did not get a ton of directed education in, right? Like, so I think that is, that's a completely legitimate and widely accepted view of what these cells do. There is actually some really interesting neutrophil literature that's relatively new, although there's one particular faculty member here that has been thinking about it for a long time, that looks at like the polarization states of neutrophils as they enter into tissue, and has found that there can actually be independent polarization states of neutrophils. So there's like a net producing neutrophil polarization state, and also there are other mutually exclusive polarization states. And what's interesting about that is that they found that that polarization state can propagate in time beyond the lifespan of a single neutrophil. So what they find is, you can get engraftment of neutrophil generation 0. That neutrophil generation 0 can differentiate functionally into some kind of specific neutrophil. And then even though those neutrophils continue to be short-lived and there is no memory, they can actually inform the generations that come after them, which is a really interesting thing to think about. We don't obviously need to get into that here, but yeah, you're absolutely right. They are the fast actors. They have a tendency to show up, to do their thing, and then to go away unless there is some other weird chronic stimuli that they're exposed to. And so, yeah, I think the question is, like, how do they cool it?
Right.
What's the balance here between, chasing around your own mob, like literally chasing your tail, and being present, being mindful of the larger tissue, bring in, mindfulness and immunology, which comes in periodically, actually, usually comes from Kevin. So you're welcome,
Usually,
everyone.
yeah. I don't know if I don't know if I need to object to your particular characterization here, or if I'm just going to let it go. I think I'm just going to let it go.
All right, done. So our mindful neutrophils need to find. I'll just keep pressing. So they need to find
I mean,
some
to be
sort of.
fair, the mob-like state of a macrophage is very similar to what it feels like internally when you're meditating and being mindful. Never mind. Carry on.
All right. We've digressed.
I mean, if we only digress this much, this will be a very focused audio.
That's true. Usually it's much worse than this. I'll drag us into what's good in 2026 in science later. But
cool.
So like you said earlier, the methodology here is actually quite simple. There are four kinases that phosphorylate G-coupled protein receptors in neutrophils, right? So presumably, one of those kinases might have an impact on the migration patterns of neutrophils, specifically one of the ones that responds to CXCL2 or these swarming ligands. And so they did what one would do, I think, routinely 4, 5, 6, 7, probably 7 years ago when this paper was being conceived of, which is they made a mouse for all of those kinases, right? So now one might imagine a situation where you would CRISPR or do something similar, but maybe you'd still use this approach. You're going to eventually have to do the specific knockout anyway later, but they basically go in, they knock out these different kinases, and then they ask the question, how well do they move? And so we're right into the microscopy immediately. You can do this in a dish. You put neutrophils on one side, you put your chemotactic factor of interest on the other side, and then you hit go, right? And you literally with a microscope sit there and you watch them move around towards that stimuli. And sure enough, your wild type neutrophils that have all of their kinases intact move pretty well towards the stimuli. But interestingly, if you look at your G-coupled protein receptor kinase 2, specifically 2 and not 3, not 5, not 6, but 2 knockout mice, knockout neutrophils, what you find is that they actually migrate at a much faster clip. And so they essentially use a number of different places in this paper, the ratio of migration from the knockout, the kinase 2 knockout animals to the wild type as a ratio. And so what you're seeing in figure 1A is actually, I think, the data from figure 1B, which is the only quibble that I had with the whole thing. It felt like figure 1B should come first. But it does seem like, you get a lot more motility in your kinase receptor 2 knockouts.
Yeah, I do want to just clarify that you said they move at a much faster clip. And I think it's worth distinguishing. That is true also. But it is worth sort of especially in a situation like this, separating out the speed at which the cells move
Yes.
and the overall distance that they travel, because they're not
Yes.
necessarily traveling in a straight line. And you can
Correct.
see this in the tracks image in B, where they're sort of doing these squiggles. They're definitely directed. It's not just a random walk where they're diffusing sort of randomly. In this case, they are both moving faster, and also they are going further away from their sort of home base. So this first one is looking at the displacement ratio. How far do they move in the
Yes.
time allotted, as well as the speed later on in D. How
Mm-hmm.
fast are they actually moving when they get going?
Mm-hmm. Yeah. Yes. Agreed. So yeah, they're actually measuring 3 independent units here: displacement, speed, and straightness. The first time I read it straightliness, and I like straightliness better. It seems
Okay.
British and more proper. Um, so interestingly, and this is, I think, the meat of what we're going to see later, right? This is what really is happening here. In the late phase of that experiment, and this is another time when it's a good thing that everybody was looking at kinetics, which you should always look at kinetics because kinetics are important. If you look at the late phase, so not the first 90 minutes, but the second 90 minutes of this experiment, this is where you see the major difference, right? So by 90 minutes, functionally, the wild type neutrophils have stopped moving. But at 90 minutes, the kinase 2 receptor knockouts are still moving almost as fast as they were in the first 20 minutes, right? And so we see that a little bit. You can see it visually in D, but the velocity reporting out of E, so basically looking at instantaneous speed, um, you can see, have they slowed down? Yeah, maybe a little bit, but really not much. They're actually moving pretty much as quickly as they were at the beginning of the experiment, whereas the wild type cells have pretty much stopped. There's a clear morphology change. Um, and what they show, importantly, is that, this is not happening to all stimuli, all neutrophil stimuli, right? What we were talking about before becomes relevant here, where we are looking at um, essentially the difference between the receptors that are responding to autologous chemokines, right? So CXCL2 and LTB4, these are the chemokine and uh, migration factors. LTB4 is not actually a chemokine, but actually, no, it counts as a chemokine still, doesn't it?
This is not. I don't care.
It's a motility lipid. Anyway, it doesn't matter. We can cut that out. But essentially, what you're seeing is the difference here between factors that are produced by the neutrophils themselves, including CXCR2 and LTP4, and other chemotactic agents that the neutrophils respond to in that initial inflammation. And I used C5a as an example before because I knew it would show up here, right? So you don't necessarily lose the ability to sense C5a still, right? You're gaining that additional motility, specifically in the case of these swarming phenomena. And so you ask the question, can you quantitate that by just looking at activity? You would expect that if this kinase is phosphorylating the receptor, maybe if you hit the receptor the first time, it would respond normally. But then if you hit it the second time, maybe it wouldn't anymore. Maybe if you hit it a third time, it would decrease even further than that. And so that's essentially what they're doing in H and I. They're looking at sequential responses to LTB4 and CXCL2 in the kinase knockout versus wild type animals or cells, excuse me. And what you see is that indeed the first time a wild type versus a knockout cell is hit, you have about the same calcium flux, which is just a measure of sort of signaling activity broadly in the cell. But by the time you get to the third hit of either of those signals, your wild type cells really aren't responding at all. They've essentially desensitized themselves, whereas the kinase receptor knockouts continue to maintain that sort of high level of calcium flux.
Yeah,
So
I
I
think
think,
another piece that they don't really they don't really get into it a lot, but I think it's relevant here, especially as we think like what could be the mechanism, like what is um, GRK2 doing here. I I really like also seeing in those curves in H and I that it's not just that subsequent stimuli are still active. It's also that the off rate
Mm,
of signaling is delayed by a lot
that's true.
in a couple of cases. So the area under the curve, like you imagine you might have like a short spike or you
Hmm.
have a spike and a sort of drawdown. Even in the wild type, there is a bit of a drawdown, especially after the first signal.
Hmm.
But the that decrease in calcium activity over time is much lower. So you have a higher area under the curve. You've got continuous signaling happening even after just one hit of the
Hmm.
stimulus,
Yeah.
which I think is also quite relevant.
Yeah. No. Agreed. Agreed. And that seems to be more pronounced even in the LTB4 versus the CXCL2, although still present in the CXCL2 on that first hit. So then you can watch one of those really pretty movies that Kevin was just mentioning. Embedded directly in the paper is that movie from Figure One, right? So these are essentially the tracks from Figure 1B, functionally. So
Yeah.
you can go and watch those neutrophils fly around in a dish. Do it out.
All right. So for movie one, this is just basically visually showing what we were talking about. So you've got wild type on top, you've got the GRK2 knockout on the bottom, you've got a stack of neutrophils um, over on the left, and you can just watch them start to migrate. They start going faster with the GRK2, and over the course of, I think this is 3 or 4 hours or 3 or 4 minutes. I can't
I think it would
remember.
have to be 3 or 4 hours based on the
Okay,
time
3 or 4
course on
hours.
the
They're
first.
they're going crazy, and then they show comparing to the other knockouts. Basically, the 4 times GRX knockout is the same as the GRX2 knockout, so you're getting no additive value from those other GRKs. That's it.
Cool. So then, the question is, I think, can we pull this into a slightly more biologic setting, right? Do we see these same sort of migration changes, maybe specific to swarming rather than recruitment? And can we do that in a way that makes reviewers think, that's probably happening in a person? Um, so they start with this microprinting assay,
This is cool.
which is interesting, right? So they're basically taking Staph aureus, he killed Staph aureus, and they're printing it as if it's a microarray. And they're basically putting that microarray on the bottom of a dish and then dropping neutrophils on top of that dish. And they're like, how well do the neutrophils get to the spots? And the answer is pretty well.
Very well. Very
Yeah,
well.
they don't mind crawling along that microarray essentially, and they do find the he killed Staph aureus, and the kinase 2 receptor knockouts do a better job of it. They tend to um, displace, basically, not fully, but they are highly competitive in comparison to the wild type neutrophils, and you see that quantitated in B as a accumulation index ratio, which is functionally, the number of neutrophils of the GRK,
Right. Yeah.
the
So to be
kinase
clear, I don't
two.
I don't know
Yeah.
if you explain this exactly. They're in this assay in the same
Uh, yeah.
dish, they're including both wild type and
Yes.
GRK2 knockout neutrophils that are differentially labeled. So you're basically watching and seeing if the index was 1, basically the wild type and the knockout neutrophils would be equally likely to accumulate on the staph spot. Um, but they're seeing actually the knockouts are accumulating more densely around that spot.
Right. Right. Interesting that they didn't show that image. Like I would have liked to have seen the differential neutrophils on the spot, but I'm certain based on the
Yeah, I think
quantitation
I
that
think that's in the video in movie 2, actually.
was it. I
Yeah.
must have missed that. Oh yeah, yeah, yeah. No, that's tissue injury, right?
Uh,
Anyway,
Maybe.
doesn't matter. So
Yes, you're
regardless, the neutrophils do find microarrays printed with heat-killed Staph aureus, and the ones that don't have the breaks, that have the continuous signaling, that have the higher signaling, seem to find those spots more efficiently. Which again
or
is
they
great,
persist on the spot
or
more,
they persist on the
which I
spot.
think
And
is actually mechanistically, I think. We
could
can get into
be.
that later.
Yeah,
I
yeah.
think, but I think that distinction matters, right? Like
No, I think it does. You're right. You're right.
so there is a difference between if there's some motility barrier, like if the neutrophils aren't able to move, that would be one thing. But I think what we're going to see is that this is actually not about the neutrophils' ability to get to a place. It's about the neutrophils' ability to get away from the place, essentially,
Yeah.
to stop the swarming behavior, right?
Yeah, let's come back to this. It'll be interesting at the end of this, after we describe it. Let's put a pin in
Mm-hmm.
figure 2A and come back to it. Persistent swarms in C, right? So now we are moving from an in situ, or not in situ even, in vitro assessment of neutrophil migration.
Mm-hmm.
And what we're asking is, do we see this same sort of difference in motility in an actual animal model? And we said before, there are two different versions of neutrophil motility that we're actually interested in here. The first is recruitment. Can a neutrophil get from the blood vessel into inflamed tissue? The second is, can they swarm effectively? Can they respond to each other's signals and find a concentrated location, right? And so the difference, and I like this actually, the difference between D and F is actually the measurement of those two different things. The first is measuring the entry of neutrophils into that inflamed tissue. And you can see from the tracks and you can see from the quantitation off to the right, there really is no difference. It does not matter if you're a wild type neutrophil or a receptor or a kinase knockout neutrophil, you can get into the tissue. That's not a problem. And what's interesting is that's not the case as you move into this sort of self-aggregation type signaling, that will allow those neutrophils to then, continue to find themselves, continue to find the mob. And so essentially what you're doing here in both models is you are taking a laser and you are firing that laser. This is probably a multiphoton, firing that laser at a high enough power that essentially you are creating tissue damage with the laser. And then what you're watching for is the neutrophil response to the damage you just did on the laser. And I would remind everyone that these experiments are done with the animal under complete general anesthesia. So these animals are asleep and ultimately will not recover from these experiments. They never wake up from these
Mm-hmm.
procedures. But you can think of the type of injury here as like an actual sort of mild to moderate burn injury functionally. So what we've got here is a quantitation in G of the relative abundance functionally of the receptor knockout animals to wild type animals in these aggregates. And what you see is at the very beginning of a 45-minute movie, they are roughly the same. But as you go over time at 45 minutes, you've essentially started to swamp the system almost 2-to-1 with the knockout neutrophils over the wild type neutrophils. They did this with the other mice because I'm sure they had made those other mice. And even though they had seen that there were no changes in the in vitro, I'm sure some PI similar to you or I said, why don't we just do this experiment 14 more times in the other kinase knockouts?
Or it
And
was a reviewer request. I don't know.
maybe.
I mean, I think it is useful because again, like when you are measuring, there's a bunch of different components to the movement. And if you're just measuring one thing, which is accumulation, that could be the result of impaired getting to the space. it could be the result of faster exiting the space,
Mhm.
or it could be the result of a knockout having a slower egress from the space, right? So like each of those things could have a different mechanism, could in principle have a different signal. Um, if, for instance, if GRK5 in this particular thing all of a sudden had a much higher accumulation index, we would be saying like, oh, what a coup that they figured out there's actually two different components. In this case, they didn't see that. All of the other knockouts are identical. Um, though interestingly, why didn't they include wild type here? Uh,
Well, it's
but
relation to wild type. Everything is in
that's
relation
true.
to
Oh,
wild
that's
type.
right. Because
Yep.
one is wild type. Okay,
Yep.
that's true.
Yep. Yep. Yeah. So no
Fair enough.
difference in comparison to wild type of the other four or the other three. And then they do something that I actually think was pretty important. It's going to be sort of overlooked, I think, in this paper, which is that they did the same thing, except they did it in eosinophils. And so eosinophils also swarm. They also rely on these same sorts of chemotactic crosstalk factors. So what they do is they go in vitro and they use C. elegans. And they basically ask the question, do we see these same kinds of changes if we mess around with those kinases in eosinophils? And the answer is actually yes. So it seems like beyond this being a neutrophil specific thing, what we're looking at here is a way to regulate this sort of swarming behavior in these highly motile cells,
Yeah, and
which
just
I liked that.
yeah, I 100% agree with you. So eosinophils just they're a granulocyte. They whereas the neutrophils are these phagocytes and also can release a lot of cytokines. Eosinophils are kind of like the Th2 branch kamikaze or mob berserker type cell. They respond to things like worms and allergens. So yeah, I think I agree with you that that is a very cool experiment. They don't spend a ton of time following up on that,
No,
but it is very nice to see that it is exactly the same, kind of thing with this particular knockout.
agreed.
Quite cool.
Agreed. And then
So
we move and we see it happen. So movie S2 is again
yeah, so movie
embedded.
2, I'm just jumping back here again. I'm gonna splice these in to the video for those of you, but I will try to narrate for those of you that are doing audio. So here we're just seeing the movies of what they've quantitated here. You've got the tissue injury, and you can see the green neutrophils or red neutrophils coming in. The red is wild type, the green is the knockout, and you can sort of see them kind of competing to get into this space. So they have the laser induced injury. And again, we're watching sort of a comparison between the two different backgrounds, and that was just in the course of 16 minutes. And they also show the movie of them coming around to the larva, which is pretty cool. Pretty cool to watch.
Yeah. Yep. No, pretty neat. So then we move on to how might this play out? So then the question becomes not so much, whether there are deficiencies or changes in vivo, but in the specific case of sort of complex chemokine gradients, right? So, you have this kind of very directional approach that we've taken up until this point. So all of the models that we've used, um, although I would argue maybe that the heat killed microarray printing is a little bit different, but the in vivo model that we were just using is a very directed kind of injury, right? We have really one source of chemokine gradients. And one of the things that, we noticed is that it's this sort of persistent ability to respond to the chemokine gradient that really is changing. So, what does it look like if we start throwing curveballs at these neutrophils, essentially, right? Because you can imagine in a complex infection, you might have an initial site of infection, you might have bacterial trickling in from another site, you might have, multiple sites that all need to be controlled. So, how does this system play out in a more complex way? And so, they do a relatively simple version of that up top, which is to start by just asking the question, what happens if you just like create a second gradient a little while on? And so what they essentially have is a in vitro system set up where we've got two independent chemokine gradients. The first is essentially due north. according to their diagram. And they basically just let that sit there. But then 3 hours in, they add a second gradient, which is 90 degrees off to the right. And they ask the question, how well did the neutrophils find that second gradient, right? And so one might guess, based on the calcium flux data that we saw earlier, that the neutrophil would be able to, in a wild type animal, follow that first gradient, but maybe not follow that second gradient because it's probably already shut itself down. Whereas the knockout animals, those neutrophils might be able to continue along those lines, and that is functionally exactly what you see. So what we see in Figure A is that when you introduce that second gradient at a 90-degree angle, the wild type cells functionally can't follow it. They don't follow it. Presumably because that receptor has already been heavily phosphorylated and shut down. So then we're looking for a system, I think, that that is more infection-like. I think really what we're doing here is just introducing another model and seeing how the system responds.
Yep.
To be honest
We
with
have
you,
fancy technology, do all the things.
there's not much intellectual distance between Figure 2 and Figure 3 here. One could argue that they're kind of doing the same thing, but that's okay. This is a little more complicated now. Again, we are looking at the subcapsular sinus of a lymph node. These authors have experience with infection modeling that kills subcapsular sinus macrophages and results in these kinds of gradients and results in neutrophil swarming, which is, I'm sure, the backstory of how this paper came to be. And so, they ask the question functionally, what happens when these subcapsular sinus macrophages start dying and getting infected with these different bacterial infections? And the answer is, once again, we've got, we've functionally got a system where the receptor knockouts are capable of aggregating at a higher density than our wild type systems. Okay, yeah. So in D, what we're looking at that same swarm aggregation assessment of the knockouts versus the wild type. In E, what you're actually looking at is scanning breadth, right? So how much distance is really being covered. And I mentioned that we would get back to intravital 2-photon microscopy. This is in a popliteal lymph node with foot pad injections. This is exactly the experimental setup we were talking about in the last episode. So they would have anesthetized this mouse, exposed the popliteal, injected those animals while the scope was running, essentially. And so you're looking at the tracks and the breadth of those tracks. And then they did what I imagine they assumed would be the clinching experiment, although I'll bet that this probably came at the very beginning.
And before you get into this, though, I think it's worth saying. So here we're getting towards the end of Figure 3, and we see that these knockouts, the neutrophils move faster, they aggregate around damage and infection more. They are able to change direction in response to a new signal. Everything leading up to this point, I would be like, these knockout neutrophils are better neutrophils. They're faster, they mob up more, and they can adapt better. Like these are better. So now I'm going to infect these mice and watch these neutrophils are going to kick ass.
Mm-hmm.
That's where I was when I got to F.
Excellent. Good. Well, they had you then, because the answer is they're worse. Their neutrophils are worse en masse. They do not inhibit bacterial replication as effectively as wild type neutrophils, which is interesting. And then they go through like a whole litany of things, which I imagine are all reviewer associated, right? They basically say, reactive oxygen species are still there, net formation is still there. Like basically what they had to do, I think then, was test all of the molecular functions of neutrophils to make sure that it wasn't the migration. The migration was better, but maybe they can't make nets anymore, right? So
right, exactly.
So they go through that.
This migration looks better on the metrics that we think are good. So if they're worse at clearing infection, I think it would be reasonable to assume, oh, we've actually just fucked with something that isn't movement.
Mm-hmm.
So we got to go look at all the other stuff.
Yeah, exactly.
But all the other
So,
stuff is fine.
yeah,
So
all the other stuff is
apparently
fine.
this movement stuff is bad.
So, yeah, the movement itself seems to be a problem. And so what do you do there? Well, what you do, first of all, is abandon ship immediately on the intervital multiphoton microscopy, right? Because this is exactly the kinds of questions that people think that they're capable of answering with that kind of thing. And in reality, no two surgeries are alike enough to actually do that experiment well. And so what they do is they say, well, we've got this interesting phenomena that's going in the subcapsular sinus and a failure to capture or a failure to control bacterial growth. So what if we tried to make a subcapsular sinus in a dish? And they kind of do that. Kinda. I mean, it's hard to make a subcapsular sinus in a dish. I would quibble a little bit with the fact that they have layered macrophages on the bottom and then put neutrophils on the top and called that a subcapsular sinus. Um, I would at least have put lymphatic endothelial cells in the mix on that if we were going to. But regardless, there's
How
like
doable is
17
that? I
cell
mean,
populations.
yeah,
What's
exactly.
that?
This is already very
Yeah.
doing this kind
Um,
of stuff in vitro is hard.
It
Let's
is.
be real here.
It's
It's hard.
totally and this is totally fine. I'm just bitching. So basically, do you still get the same phenomenon? Can you get an increase in clustering in this mimic, essentially, in the receptor kinase or in the kinase knockouts? And the answer is yes. So seemingly in this culture system, um, we do still see seemingly better aggregation, right? So we're recapitulating that, which is good. Um, and also, we're seeing more free bacteria, right? So what they're showing in the back half of C is, yes, you have more aggregation in the beginning, but also you seem to have more free um, free bug, which is interesting, right? So you're saying that the aggregation is better, but the control is worse. And so
Yeah.
then they
So and it's not a huge difference in either case,
no, it's
but
not.
so one of the things, I mean, there's a couple of things that could be going on here. So one is like if they're just flying past, maybe they're like, "I'm gonna get you, Pseudomonas!" Ah! And they just go right past, and they're not actually able to grab. So even though they're aggregating, if you're looking at a gross level, they're all sort of aggregating around in a particular place. But we also know they're moving faster, and so if they're moving faster around the bugs, maybe they can't slow down enough to actually, engulf a bug. We also saw that there's morphology changes way back in Figure 1. And so I wonder if their cytoskeleton is just like super active. They're not actually able to like form the pseudopods to actually grab onto the bugs because they're so focused on continuing to move because they can't actually slow down the chemotaxis.
Yeah, and what we're going to find is that they don't answer that question. And this is the one quibble that I have with this paper. I think that this is a good paper. I think that the biology is interesting. I think this twist we just uncovered is very interesting. I think it's all good. I think I am surprised that that last question that you just posed didn't need to be answered in order to get
Mm.
this the last bit into science. You could imagine some like really high resolution imaging uh, showing uh, the difference in trying to engulf these bacteria between these two conditions. Like there are a lot of imaging based things that one could do here, and I'll show you. We'll talk through what is shown. So what is shown when you track individual cells in a neutrophil cluster, what you find is in the kinase receptor knockouts, or the kinase knockouts. I keep doing that. In the kinase knockouts, the tracks are more chaotic. They do a lot more moving right? It sort of has an ADHD phenotype. They're there, but they're not like staying there, right? They're not super focused. They never really come to rest,
It's the
right?
HD part of
Yeah, the hyperactivity
ADHD.
component, exactly. So they're like, they get there, but then they like can't quite sit still, right? They just keep moving around. And you see that, yeah. I mean, that honestly is the biggest thing that jumps out to me, because then they get into a phagocytosis assay, and indeed they are not phagocytosing as well,
And they
despite
just,
being
on the previous point, they come up with another coefficient. I mean, this paper is littered with new coefficients. A rest
yes,
coefficient,
right.
which is just stopping, right,
Yes,
is much
yeah,
lower
the number
in the
that stop
knockout.
functionally.
Yeah.
Yeah, and so certainly they're picking up less bacterial load, right? That's undeniable. They saw that back in B, right? So G appears to be somewhat similar. I'm not quite sure what the difference is there, except that it appears to be a quantitation of B. Um, I don't know. Let's scrap that. So in
I
G,
mean, I think it's just the difference. The difference is like in the cluster versus just
outside of
overall,
the cluster.
right? They're sort
Yeah.
of focusing in on the clumped.
Okay. Yeah.
Is that right?
It could be. It could be. But then we get to H, and we're looking at percent bacterial containment, and they appear to be drawing two-dimensional boundaries in three-dimensional spaces, where they're saying that there are these large, it seems like larger clusters of PAGFP, which is the bacterial model they're using. And what they're saying essentially is that, yeah, the neutrophils are clustering well, but they're not actually encapsulating.
Yeah, they're not actually surrounding where the microbes are.
Right. And so I think what they're getting at is like a higher order organization of neutrophils that they believe is required to actually contain these things, which is like a bee thermal attack model. Where, have you ever seen the bee videos where they attack
Mm-mm.
a hornet,
Oh
like one of
yeah,
those murder hornets, where
we just tried to
they
watch this. There's a new National Geographic uh, show about bees, and we tried to watch this with the kids.
Mhm.
Um, they got freaked out by the wasps,
Yeah.
but they do show this. They
Yeah.
just like cluster on them and start beating their wings.
Yeah,
It's cool.
exactly. They basically, it's the cumulative effect of all of the surrounding organisms, right? And so I think what they're arguing here is that one neutrophil is not enough, that you actually need a cluster of neutrophils that completely surrounds this bacterial focus. And I have to be honest with you, I don't know of a clear mechanism by how that would be explained based on the cell biology of neutrophils that I am familiar with.
Yeah.
It seems like the phagocytosis makes sense to me, right? Like if they're less capable of phagocytosing, that makes sense. I don't conceptually understand the containment argument, except that
Yeah.
you could argue that they're literally flagelling themselves away because there's not like a solid cell mass. But even
I
then, I
think
don't quite understand it.
my interpretation is more that, like, if you imagine, again, thinking back to the mob example, right? So let's say you're trying to make a police line to enclose. Let's say that we are in favor of our current government and their response to protest in places like Minneapolis and LA. What's good in science in 2026, Matt?
Nah,
We won't talk
we're
about
all
immigration raids, but
no.
imagine you're trying to make a police line around a group of protesters. You can't just have individual cops with riot shields just like running around trying to hit individual protesters. You've got to have they've got
Although
to
we've
slow
seen
down.
that,
They've got to make a lot, right? But that's going to allow a lot of protesters to escape, right?
Yeah.
So in this analogy, If you are neutrophils and you're trying to make a cordon around some Pseudomonas agitators, somebody's going to clip this out of context and make me sound like a fascist. When dealing with infection, we want to be more like fascists. No, I'm not going to say that. Fascism bad in society. In infection
But not
control,
in neutrophils.
maybe not so bad. But yeah, so if you want to cordon off a particular space, you need to be coordinated, you need to slow down, you need to cluster, you need to be organized. If individual neutrophils are just flying off all over the place, they're not going to be able to hold the line, essentially, right? This is the difference, again, going back to like trying to throw in all of these analogies here, going back to like thinking about like a military battle or something. You just have berserkers that are just running around smashing things, as opposed to like a disciplined line of slow-moving infantry that are going to like have a shield wall. It's like there are benefits to that maybe in like early stages of battle, but if you want to actually contain the infection here, you really want to have the ability to slow down and organize.
Yeah. Slow down and organize. That's what's good in science 2026. Slow down and organize.
Oh, good transition, Matt. Good transition.
Yeah. Yeah.
That's the paper.
Yeah,
We skipped the third and fourth movies, but honestly, they're a little bit tough. When I look through those movies, they're a little bit harder to see what is actually being shown. And you can see that in the quantitation. It's a little bit less obvious what the difference is. You kind of need the quantitation and the statistical test to tell you that they're super different in these. I believe it's different. And the infection model in 3F, I feel like that's the crux of this paper. And that doesn't lie. Like you give it an infection, and these neutrophils can't contain it as well as the wild type. Which again, like that surprised me, but I think it's a very cool result.
Yeah. No, it is. I enjoyed this paper. I think that we've been hitting the molecular pretty hard recently. I think going back to some good old fashioned let that sell off in addition, watch it go for a little bit.
Yeah.
It was a nice transition. So, and I actually
I'm
gonna
to be
skip
able to do experiments like this.
Exactly. I'm gonna save everybody the what's good in 2026 segment because it feels like a nice pleasant way to end our discussion.
I dig that. In that case, this has been Audio Immunity. I'm Kevin Bonham, joined by Matt Woodruff. We have been slowing down a little bit on our recording schedule. Both Matt and I have been quite busy. It turns out when you are new PIs and you need to be constantly writing grants, you have less time for podcasting. This is going to be especially true next month. I'm trying to submit like 4 grants in the next 2 months, which may or may not kill me. If this is the last episode of Audio Immunity, forgive me. But
now, so I'm sorry, I have to interject. So that was actually what I was going to talk about for What's Good in Science 2026, which is this new conspiracy theory that foreign agents are murdering random scientists with no clear connection to each other. So if Kevin is dead, you'll know that it's because there is a foreign cabal that is randomly killing really anyone, because there are interestingly random biologists on that list.
I did not know about this conspiracy theory, and I don't know that I wanted to know about this conspiracy theory. You'll have to send me a link. Or will me knowing about it make me more likely to succumb? I have no idea. We're going to try to record at least once a month. Even in the crazy busy grant submission deadline, we're going to try to record at least once a month. If you'd like to support us, please go to Substack, substack.com/audioimmunity. Should spell it AUDIOMMUNITY. Website audioimmunity.org, which is still active, although I haven't updated it in a while. I think we're going to have to just redirect it to the Substack eventually. What else was I going to say? Oh, because our recording schedule is slowed down, like I can understand you don't want to necessarily give the same recurring donation on a monthly basis. Send us an email. I'll send you a discount link. You can give us $1 a month. You can give us 50 cents a month. Honestly, any amount of support is super welcome. We just had somebody sign up at the $5 a month rate yesterday, which is super cool. What else? You can send us an email, contact@audioimmunity.org. You can find us on social media, on Blue Sky. Those links are on the website. And the music at the beginning and the end was composed by Rachel Rannick. We'll see you next time. Hopefully, without being murdered in the interim.
Here's to that.
Sometimes you get